So we just talked about how antibodies are high molecular weight proteins produced by the B cells in response to a specific antigen. They are crucial in our body's self-response against any type of offense, and they're usually represented as a Y-shaped molecule. The antibodies have 2 heavy chains and 2 light chains. The 2 light chains comprise the FAB region of the antibody, while the 2 heavy chains comprise the FC region of an antibody. On the FAB region, we also have the variable region of the antibody, which is responsible for the antibody's specificity against a given antigen. All antibodies have the same basic structure or arm, which is comprised of the 2 heavy chains and the 2 light chains, or the FC and the FAB regions. While all of them have the same basic structure, they can be differentiated in different isotypes based on the number of arms they have. So since the number of arms change and they have different structures, they can play different roles in the immune system. Antibodies are mostly found in the blood serum. The blood serum is the fluid part of the blood minus the coagulation factors. There are 5 classes or isotypes of antibodies in the mammalian blood serum. They are named Ig, after immunoglobulin, plus a Greek letter. So we're going to have alpha, IgA, delta, IgD, epsilon, IgE, gamma, IgG, and finally mu, IgM. Each one of those isotypes is present in different parts of our body, and they have different roles in the immune response. IgG is the most common form of immunoglobulin, and it's correlated with resistance to an infection, while IgM is correlated with an active infection. IgA is commonly found in mucus secretions as well as in milk, whereas IgE is commonly related to allergies, and IgD, which is the least abundant of the immunoglobulins, is typically associated with the IgM expression and is found in respiratory and gastrointestinal systems. For example, if your doctor is wondering if you have a current infection, they're going to ask for a blood draw and look at your IgM levels. Whereas if they're wondering if a vaccine is still working, they're going to look at your IgG levels. Now, it's actually very interesting to know that different animals can actually produce different isotypes of antibodies. And looking into the different isotypes of antibodies that different animals express has actually shed a light into their evolution. For example, chicken produces a completely different type of antibody, which is called IgY. In addition to having different isotypes, a lot of animals also have different subclasses of antibodies. Those subclasses are characterized by different modifications in their heavy chains. For example, humans have 4 different IgG subclasses, IgG1, 2, 3, 4, while mice have 5 IgG subclasses, IgG1, 2A, 2B, 2C, and 3. Now, using the different isotypes and subclasses to our advantage, that allows us to multiplex several primary antibodies in one Western blot protocol. We're going to talk more about that later. But first, let's focus on the bonds between the antigens and the antibodies. The bonds between the antigen and the antibody are non-covalent bonds, and therefore they are reversible. Some examples of those bonds are hydrogen bonds, hydrophobic interactions, electrostatic interactions, and even van der Waals forces. Keep in mind that each antigen has more than one epitope, and therefore more than one antibody may be binding to each antigen. The affinity of an antibody is the strength between one particular epitope and its paratope, while the total strength of all the epitopes together with the paratopes is the antibody avidity. Antibody specificity is how well can an antibody discriminate for that particular antigen of interest. Keep in mind that because these are non-covalent bonds between the paratope and the epitope, those bonds can be reversed by experimental and physiological conditions. So since we're talking about experimental conditions, let's think a little bit more about how your antibodies are used on your Western blot protocol. So if you're ever curious as if it really matters if you use PBS or TBS on your protocol, or if your buffers are at a pH of 7 or 7.2, or Or even if you incubate your antibodies at room temperature or at 4 degrees, the answer is yes. And the reason for that is that all of those conditions can affect all of the different biological and chemical reactions, including those between your epitope and paratope.